This one without a picture, but you get the picture anyway. So we're going to talk about different ways of making phylogenetic trees. And remember that all phylogenetic trees are things that are calculated. If we make them right, they should reflect what we understand about taxonomy and the rest of what we understand about development, for example, and even the fossil record. If we do them badly, they probably won't, and we've got to look really closely to make sure that everything we're doing makes sense. So with these phylogenetic methods, the only real data that is in there is going to be at the tips of the branches. So that's where you find basically the sequences that we have. All of the nodes, and nodes are places where one or more, two or more branches join together. All of those are places that are calculated. And if our calculation is doing a good job, those should roughly correspond. Like two species should join at a genus, two genera should join at a family, etc. Something like that. It's never going to work out exactly. I mean, life isn't broken down into exactly that number of steps. And to some extent, it's arbitrary where you say the genus line is versus the species line versus the family line, something like that. But yeah, this is how we get that data, and this is how we basically get into it. So let's go. All those nodes are called operational taxonomic units. So that just means a point where two of these little branches join together. And all that means is that we are hypothesizing a common ancestor of those two things, which has a genetic distance, or whatever distance we're measuring, of whatever the graph says from those two species. And remember, you count the distance up to it and then the distance back down to the other one to see how far apart those two species would be. So let's look at some methods. All right, we'll start with UPGMA. So UPGMA is unweighted paired mean grouping algorithm, which, there we go, unweighted pair group method with arithmetic mean. Yeah, sorry, I had a couple of those switched around, but means the same thing. So these are going to be in the, we can basically break these down into different categories. So some of these methods will try to cluster based on distances. Now, there actually are clustering algorithms that will cluster based on characters. Those are not going to be molecular phylogenetic things. Those are going to be like if you have a tree of dinosaur bones and you measure all the little distances between all the little features on them, you can make a family tree out of those numbers, or out of numbers that say, you know, does it have an eye hole, yes or no, you know, 1 or 0. So those exist, but ultimately those are only as good as the methods used to reconstruct them, and the only way we can really test whether those methods are working is with molecular data, which means DNA, protein, RNA, stuff like that. So that's what we're going to stick to. And I guess we can then assume that if these methods work well enough for RNA, and if no other data is available, then we can say, well, maybe it also works for dinosaur bones, and probably that's the best we can do. All right. So the main goals here are, some of these will try to cluster different taxa together. So UPGMA will try to cluster things. It basically tries to draw like a circle around the two nearest things and say, okay, anything inside this circle belongs to this operational taxonomic unit. Neighbor joining is kind of neat. It starts out looking like, they call it like a star diagram. And you're going to take the two branches that are closest to each other and say, okay, those two are going to join each other. And now where does that join onto the group? And then you keep doing it and saying, okay, now that we've got these two separated, which two are the next most closely related? And you group those. And so every time you find the nearest neighbors and you join them together. We're going to look at methods that maximize likelihood. So that's going to be stuff like Bayesian statistics are going to do that. We're going to maximize parsimony, where we try and get the shortest possible tree that could possibly explain the data. We're going to go for minimum evolution. That's another thing, which is going to be essentially what is the fewest number of changes I can make that will join these things together. And all these sort of optimality criteria, we're basically using Occam's razor. We're saying whatever's the simplest explanation, probably going to be the best explanation. At least it would be hard to justify anything else as being better just off the top. And least squares is a way of computing sort of the distance between two things. It's kind of like the way they compute the distance between a protein structure and, let's say, a reconstruction that you've made with a computer. You can say, okay, what's the root mean squared distance of these things? And that's something like that. It's a mathematical method. All right, so let's start out with these distance-based methods. So in these methods, the important thing is how long each of the branches are. And remember, the branches are different lengths if it is a phylogram, P-H-Y-L-O-G-R-A-M, and the branches are all going to be the same lengths, and the nodes are all going to be the same distances apart if it is a cladogram, C-L-A-D-O-G-R-A-M. So it's just two different ways of looking at a thing. Cladogram is showing you the rough pattern in which things diverged without making any claims about when any of this happened. Phylogram tries to do the same thing and tries to throw distance onto the top of it. If you're dealing with molecular data, usually the phylogram is the way to go. If you've got data that's only based on characters, like height, length, width, presence of spots, stuff like that, then probably a cladogram is a more conservative, sort of safer way to go, since how do you tell how long it takes to develop spots? I mean, if you get measles, I don't know, less than a week. If you're a frog, I don't know how many million years that takes, but that's the idea. So, the goal of these methods is to find a tree in which the branch length, so how long each of the branches is, will correspond as closely as possible to the observed distances between these things. So you can always calculate the distance between any two sequences. You can say, all right, we've got these nucleotides, these other nucleotides. How many changes do I have to make in order to turn the one into the other? It's a number. It can be calculated for anything, and you can do this with any two sequences, even if they are not related. It can definitely be done. For proteins, you can look at it a different way. You can say, all right, proteins, what nucleotides could make this protein? And then how many of those nucleotides would I have to change, or rather make this amino acid? How many nucleotides would I have to change to turn it into another amino acid? And so for some of these, it might just be one change. Some things you might have to do 2 changes. Other things you might have to do 3 changes to actually turn one thing into another. And so certain amino acid substitutions will be more likely than others just based on the way the genetic code is set up. And if you want to picture that, I would say pause right here, go and grab a genetic code wheel, and take a, take a particular codon, like take a methionine codon, see how many changes you'd have to make to turn it into a phenylalanine. Great. And then see how many changes you'd have to make to turn it into a leucine. But remember, there's 2 different areas of that genetic code wheel that'll specify leucine, and one of them may be closer than the other to methionine. So yeah, go have a look. And this is while we're still talking about just random changes that have no functional significance. If we're talking about changes that actually impact the way a protein folds or what a protein is able to do, like its enzymatic activity, then there's going to be a really heavy evolutionary pressure kind of sitting on top of this and really adjusting those probabilities. So it's not just going to be about what changes are easiest to make. It's going to be about what changes you can tolerate that don't make things worse or make things slightly better. Okay, so in general, these distance-based methods are not the best ones. You can do better. Yeah, you're better than that. But these distance-based methods are very fast to do, computationally fast, because we're leaving out a lot of data. But yeah, if you have giant numbers of sequences, it is very difficult to run some of the more computationally expensive things, like Bayesian calculation can sometimes be kind of rough, or there are other ways of doing this that are just too computationally expensive to work at large scales, like when you're doing an entire tree of life with all those millions of species on there. All right. And so we're going to do one more slide, then we will pause this section. Okay. So in each case, we're going to use a distance metric. So it can be a very simple one, number of amino acid changes. It could be more sophisticated, like how many nucleotide changes would we need in order to get that number of amino acid changes. It can be some kind of distance score. You can say, well, okay, a transition, is only worth 2 points, and a transversion is worth 4 points, or something like that, because that should be less likely to happen by chance. There are lots of ways to do that, and each one of these tools is going to have its own little tweak on the way that we do that. And ultimately, that is not going to be important for this course. Like, if you're designing new phylogenetic tools, definitely that's something you need to know. If you're using them, I think it's more useful just to get a feel for how these things work and where their blind spots are, because they all have blind spots, for sure. Okay, so yeah, distance is the basically the amount of difference between two sequences. So dissimilarity is a good way to say that. And similarities are useful, like for us, for understanding that we're dealing with homologs. But ultimately, the only data that really have meaning are the changes. The parts that are the same are there so that we can align these things, and we can tell which parts should be aligned with which other parts. But it's only when there's a change that we have genetic distance, and we can use that change. Is it an easy to make change, a difficult to make change? Is it a change that happens often or very rarely? That's where the interesting data is that these algorithms are going to use. And that's where the differences between the output of these different algorithms are going to be found. So yeah, distance-based methods like UPGMA and neighbor joining are very fast, but they overlook a substantial amount of information in a multiple sequence alignment in the name of going fast and being simple. So these are kind of the first methods we came up with. We can do better. We will get into those later in this particular section. But for now, yeah, that's where we'll leave it. So there you go. That's the end of part 1.